Hello! As you can see, I dressed up for class today. I didn't figure there was any point in taking this overly formal. We're just all doing the best we can in our coronavirus stuck inside situation. Hopefully you're doing that and not going out in big groups with lots of people. It's probably is important that we all try to do our part. So we're going to continue. We're skipping chapter 11, going straight to chapter 12. Chapter 12 is the nervous system. And we're going to talk about nervous tissue. If you remember, we already mentioned something about nervous tissue when we did chapter 5, the tissues. And we talked about that neural tissue. Nervous tissue is divided into nerves proper, which we call neurons, and then glial cells, which are like the supporting cells that make the whole thing happen. All right, we are going to talk a little bit about that today. We're going to talk about how action potentials work. We mentioned something about action potentials last time in muscles, and we're also going to talk about neurotransmitters a little bit, just a little. This is the bird's eye view. You know, we're just getting the big overview. We don't want to get lost in the weeds. This stuff's really interesting, and nerves they're pretty fascinating. When you think about how they work, what they do, how that information that you get from touching things and looking at things and feeling things, how that goes to your brain and gets translated. And then there's this whole other kind of secret side of it where your brain is perceiving things going on and doing things that you don't know anything about. That's with your autonomic system. That's pretty amazing too. All right, so we're going to talk about the general functions of the nervous system. So this is your primary communication and control system of the body. Okay, integrates, regulates body functions. It collects information via receptors and evaluates information to determine if a response is required. And like I said, some of those things you make in your higher levels, you know, your cerebrum looks at the information and says, hey, I think we should do this, or, ooh, that's hot, don't touch. But then there's all these other things happening at a level that you're not aware of, the subconscious, maybe even lower than that. You know, some people think that you can communicate with the subconscious and things like that. We're talking about down, low, like the way your gut functions. All that happens without your knowledge. Okay, and then we have effector systems where the nerves tell glands and muscles and things like that what to do. Okay, we're going to see how the nervous system is divided. We generally divide it into two big parts. The central nervous system, which is the brain and the spinal cord, and then the peripheral nervous system, which is all the rest of your nerves, both your somatic ones and your autonomic ones. So I'm using that term like you know what that means. We divide up input and output. So we have our sensory nervous system, where we're sensing what's going on. That's input. And then we have our motor nervous system, which is output. And then those can be subdivided. You have your somatic sensory, which is like the nerves where you perceive what's going on. You touch and feel and hear and see, all that. That's somatic sensory. But then you have your visceral sensory, where your body perceives, or your brain perceives what's going on in your body, like your pupil changes sizes based on how much light is going to your eye. You have no conscious control over that. I mean, you notice the light hurts, but you didn't decide to close that pupil up. That just happens, okay? Your blood vessels change size based on information coming to it from your visceral sensory system. So notice sensory and motor for somatic have the same name. It's somatic sensory, somatic motor, and then we have our visceral sensory and autonomic motor. So when the motor part that you're not aware of does something, it's autonomic. When you sense it, it's visceral. And you know, viscera is just, I think it's a Latin word, comes from guts, what's inside. So we talked about the two types. We have neurons and glial cells, which support and protect neurons. So notice these things about neurons. They're excitable, they're conductive, they're secretory, they have extreme long life. You know, your nerves you're born with, that's the same nerves you're going to die with, and they function all through your life. You know, even a 95-year-old man, for the most part, unless he has some kind of disease, his nerves are still intact. They're still gaining information and telling the body what to do, it's pretty remarkable. And they're generally amitotic. And what I mean by that is they don't go through mitosis. Now, an exception to that would be your first cranial nerve, your olfactory nerves. Some of those can be damaged and go through mitosis, which is interesting. But for the most part, nerve cells don't create new nerve cells once you're done developing your nervous system as an embryo. And we talked about glial cells that they support and protect. So we're going to compare and contrast muscle cells and nerve cells. And this is going to be a test question, so I'm giving it to you ahead of time. So your muscle cells also are excitable and conductive. We saw that last time. But they don't secrete anything. They're not secretory like nerves are. They usually last a long time too, and they don't go through a lot of mitosis. But nerve cells are not stretchy. If you get a big old nerve, like if you're cleaning an animal or dissecting an animal, so you pull on those nerves, they're tough. But a muscle generally is stretchy. A living muscle will stretch. Okay, so that's one of the differences there too. So we're going to talk about the different parts of a neuron being a nerve cell proper. So the soma, that's our cell body, and it has a nucleus, and of course the cytoplasm. You know, in our different cell types, sometimes they have different names for the same stuff. So instead of calling it cytoplasm, they call it the perikaryon, and that's left over from when somebody first looked at these. They thought karyon, that it looked like a kernel, and peri means around, so it was the stuff around the kernel, okay? And then Nissl bodies, these little chromatophilic substances that are all scattered through there, but it turns out we now know those are ribosomes, but some people still call them Nissl bodies. And then there's the cytoskeleton, the microfilaments and the intermediate filaments, the microtubules that make up the structure of the neuron, which stays the same because they're not changing, you know. I guess I should set this so I know about how long I'm going to talk. All right. So this long part here, they call the axon. And then these little shorter hair-looking things, these are dendrites. Okay. So when we were doing the skin, remember they had these dendritic cells down just right above the basal layer there? Well, this is a similar name, dendrites, and that just comes from a branch. I think that's all that term means. So the axon hillock is this part that changes from the body, the soma, into the axon. This is the hillock. And then of course, the stuff inside, instead of cytoplasm, they call it axoplasm. And instead of the the phospholipid bilayer even has a different name. Like remember, in muscles it was sarcolemma. So in the axon here, it's the axolemma, just the covering, the phospholipid bilayer that covers the fluid that's inside for the axons. So the ending part, we call that the telodendria. That's just the axon terminals. And then of course there's the synaptic knobs and vesicles. Remember with muscles, we talked about there's that synaptic knob, that the neuromuscular junction, we call it, that touches to the muscle. Well here, that might be what we're talking about, or we could be talking about where one nerve talks to another nerve, because the brain is just a whole bunch of neurons communicating with each other, and somehow that adds up to the amazing thing that we have in our skulls that can do all the stuff that it can do, and it's just little action potentials going from one axon, from one neuron to another. It's pretty amazing. Okay, so several videos in here. We're not going to take the time to watch the video now. I'm not even sure you could see the screen very well, but I want you to pull up the slides and follow along with me while we're doing this, and you can just pause the YouTube video, watch the other video. They're very good. They probably explain it better than I do. All right. So we're going to talk about, there's some structural classification with neurons. There's these multipolar neurons. So they have axons here, axon here, and lots of dendrites on this side. And then we have bipolar neurons, which kind of has an axon looking thing on both sides, but one side is classified the dendrite, but you can see it looks very similar. So that's why they call it bipolar. All right, unipolar, there's a cell body here, a single short process that comes off, and then these go both directions. When we talk about the peripheral nervous system, we'll see where those are. And then there's these no axon neurons. See, it's all just dendrites all the way around. There's lots of these in the brain. These are the most common, these multipolar ones. Okay, functional. I'm trying to get us through this quickly so you don't get bored watching this video. Functional classifications of neurons. So there's the sensory ones. These generally come. See, this is a spinal cord that's been cut, cross section, right? A transverse section, and this is the posterior part, and this is the anterior part. So the motor neurons come out anteriorly, and the sensory neurons come out posteriorly. But even though the tracks run up the spinal cord to the brain, they also communicate directly with each other. So there are some decisions that are made right here. What we call an interneuron makes a decision about if there needs to be a motor action taken without going to discuss it with the brain, but right at the spinal cord level. When we talk about the spinal cord, we're going to get into a little more detail on this. And sometimes there's even decisions made across here. See, the reason this is shaped this way is these are nerve tracks that go across from side to side. So this is just a cross section. I know this is confusing. This is the spinal cord. It actually runs up to the brain up here. The brain would be big right here, right? Okay. So we're going to talk about the glial cells. These are interesting to me. So you have a whole group of these, and some are only in the central nervous system, and some are only in the peripheral nervous system, and some are in both places. These astrocytes here, these are very important. They have these little perivascular feet that coat every blood vessel that goes through the brain, through the central nervous system. And this is important because this is what creates the blood-brain barrier. When I first heard about the blood-brain barrier, that sounded like a made-up word to me, but it's a real thing. And it's just an extra several layers created by these astrocytes to try to keep toxins out of the brain. Some things can penetrate the blood-brain barrier, but many things cannot, and that keeps the neurons safe because neurons are easily susceptible to toxic substances. Oligodendrites, they create the myelin. Myelin is a sheath that functions very similarly to the way insulation on wires does, right? So a wire that has good insulation can transmit the impulse better, and that's how it is with neurons as well. If there's myelin, then it can go much, much more quickly, and we're going to find out before this lecture's over why it's so much faster. And we also have these little microglial cells. See, here's one right here. There's one there, and here's one here. I don't know if you can see my pointer. You probably can't, so I'm pointing with my finger. These are little macrophages. They go through the CNS eating either in things that could cause infection or toxins that need to be taken out of there. So they function like little garbage disposals. And then we have these ependymal cells, which create the CSF, the cerebral spinal fluid, the fluid that surrounds the brain and the spinal cord. These ependymal cells are the lining inside of your ventricles, which are the watery parts of the brain, and all along the spinal cord. Yeah, I think that's everybody. So these are all the glial cells that are in the CNS, the central nervous system. Okay, and then you have your peripheral nervous system glial cells. So we have satellite cells, which are these unipolar cells that are right here in this posterior root ganglion. Remember, we said the sensory part comes in posteriorly and the motor part comes in anteriorly off the spinal cord. And these satellite cells are what make that big ganglion, that little thickened part of the peripheral nerve as it goes into the posterior horn of your spinal cord. Okay, and there's also these neurolymnocytes. When I was training, we called these Schwann cells, and I just love that word, Schwann cell, but now they call them neurolymnocytes, which is probably better not to name it after somebody. And these also make the insulation we were talking about, the myelin, that makes the conduction of a action potential much more quick. All right, so how's that myelination work? Well, when it's forming, notice here's a neurolymnocyte starting to wrap itself around a peripheral nerve, and it wraps and wraps and wraps, and every layer, it creates another layer of, it's a double layer phospholipid bilayer. And see, you can see here, there's multiple layers. And finally, it's lots and lots of layers, and that makes a really good insulation. And there's no sodium and potassium channels where there's a myelin sheath. There's only channels in the little spaces in between, okay? And those, the nodes of Ranvier, that's what those little empty spots are called. And that allows it to go much more quickly. We're gonna get into that. The action potentials go more quickly. And there's also nerves that don't have any myelin, and there's a reason why some don't. Okay, sometimes there's certain activities in the body they want the action potential to go slowly, and so that would be places that don't have them. Okay, now when a central nerve gets cut, generally that's the end of that nerve. It does not function very well after that. That's why spinal cord injuries are such a big deal. But some peripheral nerves can regenerate themselves. They don't go through mitosis and create a new cell. They create this this little sheath here, this endoneurium, that allows the phospholipid bilayer of the axon to be reproduced in that sheath. So notice it eats it up, and then it creates a whole new axon and goes back to wherever, like in this case, something connecting to a muscle. And so certain peripheral nerves can be regenerated. Okay, so as we mentioned, there's these channels. Each part of the nerve has a different set of channels. Okay, remember we talked about last time, we had ligand-gated sodium channels, that's chemically gated sodium channels, and then we had voltage-gated sodium channels. That was in muscles. Well, this is very similar here in the nerves. Okay, so up here at the top we have just these chemically gated sodium and potassium channels, and even chloride channels. Here in the axon hillock, there's just voltage gated, they're not chemically gated. And then all down the axon, what you have is voltage gated sodium and potassium channels. We're going to see how those work in just a minute. And then down here at the end, we have these calcium channels that allow calcium to go in to merge with those little vesicles and exocytose whatever neurotransmitter that particular nerve carries. So just to revisit the sodium-potassium pump here, all your cells spend a lot of energy keeping the sodium-potassium gradient perfect, okay? There's a lot of potassium in here and not much potassium out here. There's a lot of sodium out here and not much sodium in here, and that happens because this sodium-potassium pump is burning ATP all the time, trying to keep that gradient the same. And so the nerve has to keep a perfect amount of sodium and potassium on both sides for the nerve to function properly. It also has leak channels, and this seems counterintuitive at first, like you're spending all this energy pumping, why do you have leak channels? Well, the combination of the pumps working plus the leak channels, and if you watch the videos this might make more sense, that perfect combination allows there to be that exact, what we call a resting membrane potential, that minus 70 millivolt all the time resting membrane potential. Okay, and we already mentioned we have chemically gated channels too, where the chemical comes and tells the sodium or the potassium channel to open. All right, I think this is what we just talked about. So here's somebody that took this little kind of imaginary millivolt meter, and they're checking the voltage outside the cell versus inside the cell. and seeing what the difference is. So you remember in muscles, it was minus 90 was the resting membrane potential. Well, in nerves, it's minus 70. So see, there's a lot of potassium inside, not as much outside. Okay, so then the membrane potential is established by the leak channels plus the sodium-potassium pump. And they're working all the time, adjusting the chloride, sodium, potassium to keep it just right. All right, this is a pretty good video. Like I said, we're not going to watch it now. But you pause my video and watch this video, it'll make more sense. Okay, so how does synapses work? Well, this is an axon that's communicating with a dendrite from another neuron here. And notice there's different places. This is a schematic drawing, obviously. This is how they really look. There's different places where it can synapse, either right on the body, or maybe out here in a dendrite, or even here on the axon. And depending where it synapses, it affects how that nerve is going to function. There's also gap junctions between the cell to keep the charge the same on both sides. This is in a muscle here. This is smooth muscle, you can tell because of the shape. And see, there's those gap junctions to keep the charge the same all the way through the whole smooth muscle between cells. And this is what we talked about last time, where that synapse meets a muscle. This is a situation where the synapse is meeting another nerve, another neuron, but the action is the same. Calcium rushes in through these gates, these voltage-gated calcium channels. The calcium binds to the vesicles move down the microtubules and drop the contents, in this case a neurotransmitter, into the space here, right? This space in between the two nerves. And then, depending what they're showing it to be acetylcholine here, I was going to say depending what neurotransmitter it is, but it's labeled acetylcholine. That communicates with these chemically gated or ligand gated, same term, those are synonyms, sodium channel to open it up, let the sodium rush in, changing from that minus 70 resting membrane potential to a less negative number. Okay. Once it gets to around minus 55, that's when the magic happens that we're going to talk about in just a second. So some neurotransmitters are excitatory. They make the cell, the other neuron, less negative, more likely to start an action potential, or some are inhibitory. They make it less likely that it can go to an action potential by making it even more negative in the cell. Okay, so in general, a gain of sodium will cause, see it's here's minus 70, it'll make it get a little less negative so it's easier to get to the threshold where it would open. Once you watch all the videos, it's going to make more sense. And then some, it's either a loss of sodium or a gain of chloride that makes it more negative, which makes it harder to get to the point where it's going to have that action potential. All right. So here's a whole bunch of other synapses. All these other neuron axons are on this particular cell body, and some are excitatory and some are inhibitory. And depending which neurons are firing affects whether or not this body is more or less likely to have an action potential. Okay. Same idea. We're not going to get into this. Okay, so how are we going to change the resting membrane potential? That the way we would do this in this case, the way the nerves do it, is by getting more positive. The more sodium rushes in, sodium has a little plus sign, so the more sodium rushes in, the more positive it gets. And once it gets to minus 55, see, then voltage-gated channels start opening. And once the voltage-gated channels start opening, then more sodium rushes in. And notice it just gets more and more positive. So we went from minus 70 to minus 55. And once we get to minus 55, then it continues to let sodium in all the way to positive 30. And then at positive 30, all those voltage gated sodium channels slam shut. And now, once they close, these potassium channels open, letting the potassium out. And remember, potassium is positive, so then the voltage goes down. But they don't close right away when it gets back to resting membrane potential. They stay open a little longer, and so you end up with this part where we call it hyperpolarization, where it's extra negative, all the way down to -80. And then all the channels close, and then the resting membrane potential is restored at about -70. So this video explains that very well. Watch it twice. And
Neurons send signals over long distances by...
Okay, watch the video. So here we're gonna we'll overview it again, and I think you can see. Yeah, you can see it right on the board. So what you have is this, right here's the resting membrane potential. Resting membrane potential, and it's at -70. Okay, so it's just cruising along, staying, and then some sodium channels start opening because those ligand-gated sodium channels open and sodium starts rushing in, and as that happens, it gets less negative. And then at -55, okay, then the voltage-gated sodium channels open and it shoots all the way up fast because they all open. There's a whole bunch of them. And once you get to +30, then the sodium channels close and the potassium channels open. And when that happens, potassium rushes out of the cell, and then it starts getting negative. And it gets all the way down. We're back to -70, but it keeps on going because it takes a while for these potassium channels to close. It gets down to about -80. And then it returns to the resting membrane potential. Now, action potentials are either all or nothing. Either this happens or nothing happens. Nothing changes with the nerves. So once this happens, well, then that nerve can't have another action potential because it already has one going through. So we call this part the absolute refractory time. This is a time when there cannot be another signal go through that nerve, no matter what. And then this part they call the I can't remember the exact term, but it's like a semi-refractory time, right? That if you had a big enough action potential, it could overcome it, but it generally doesn't because it's hyperpolarized during that time period. We'll get to it in a second, then I'll remember. So back to remind you, action potentials are all or nothing. They spike and that's it. And they can be close together or far apart, but they can't be. You can't get another one during that refractory time. Okay. Relative refractory, that's it. This is the relative refractory time because it's hard to overcome that -80, almost -90 voltage. But here, I mean, it's already positive. Nothing can make it more positive than that. If it got more than +30, well, that just would be an unfunctioning nerve cell.
Action potentials propagate in a continuous fashion in unmyelinated axons.
Okay. So now we're talking about the difference between myelinated and unmyelinated. In myelinated, those voltage-gated sodium channels have to open all the way down the axon, and so that takes a long time. But in myelinated cells, where we have these nodes of Ranvier, it's only here that the sodium channels have to open. And so what happens is the sodium diffuses much more quickly through the axoplasm than these voltage channels can open, so it's much quicker. Okay, that last video explains very well why it's faster. What you really need to understand is that with myelin, it's faster than without. Okay, like lots faster. Like instead of 2 to 3 meters per second in unmyelinated, it's like 200 meters per second. Okay, we just explained why it's faster. So this is just general overview. Notice you can have lots of different neurons affecting one neuron, or you can have one neuron affecting lots of other neurons, just depending how the neural network is set up in that particular part of the brain or the spinal cord. That's all this picture's showing you. Okay, moving on to neurotransmitters. We can either classify neurotransmitters by their chemical structure, or we can classify them by their effects, by what they do. They can be inhibitory or excitatory. Make it more likely that we're going to have action potentials, that's excitatory, or less likely that we're going to have action potentials, that's inhibitory. Okay. Acetylcholine is the most widely used neurotransmitter. We saw it in muscles, and we see it a lot in nerves, and there's nicotinic receptors and muscarinic receptors. That's all you need to know about acetylcholine. There is acetylcholine esterase. This is a enzyme that breaks down acetylcholine when it's in the synaptic space there. And so the two ways that the acetylcholine ceases to function to open up those channels is either it diffuses away or acetylcholine esterase wipes it out. And they have drugs that directly affect acetylcholine esterase, and that affects how your brain functions. Okay, there's also the biogenic amines. Notice these are all derivatives of tyrosine, which is an amino acid. So dopa and dopamine and norepinephrine, epinephrine. You've heard of all these things because we have different drugs that utilize them, or sometimes we use them directly, that have different effects in the brain. Epinephrine and norepinephrine are catecholamines that cause that fight or flight response. All right. So how these neurotransmitters are reabsorbed can be affected by drugs. So these are, you've heard of the SSRIs, the serotonin, selective serotonin reuptake inhibitors like Lexapro and Celexa. That affects how your brain functions. These are drugs that are used for anxiety and depression with so-so results. Some people they really help, but half of people they don't seem to help at all. But this is the level at which these things work, at the synaptic level, deciding how quickly those neurotransmitters are reabsorbed. All right. Not going to get into that. One thing I do want to point out, you do have endocannabinoids. This is your naturally occurring neurotransmitters that have a similar effect on the body to marijuana, to the cannabis plant, and that's the receptor that it works at. And when you use things like THC, you're really overwhelming that system, and then you can't enjoy your normal endocannabinoids nearly as well. And also that these are lipids, which means THC lives in the brain, it stays in the brain, and you're just gonna have that floating around there for a long time. That's why your drugs test stays positive for a long time with THC. That's just another reason why I've strongly recommended you stay away from that stuff. I think that's it. That was everything I wanted to talk about. So we have some homework that's due next week on this stuff, and I'll get another lecture up as quick as I can.